Right, if we go back to the overall structure part and look at, it's not going to move for me, right, the overall roles of the kidney then, we have started on this, so this is just a review of what we did last time. We've got quite a number of functions for the kidney. We're not looking at all of these, but it's just a matter of remembering that, you know, the kidneys are incredibly important. So obviously kidney failure has a lot of implications, and we'll talk about that as well, because it's quite an interesting diversion at some point. The excretory bit at the top there, removing metabolic waste, this idea that the body makes substances it needs to get rid of. They're sort of byproducts of useful reactions. The main one is urea, and I'm not going into the biochemistry of how that's produced. Then we've got the homeostatic bits of it. Osmoregulation is the main focus for this unit, and that's in the assessment criteria that you need to understand osmoregulation. So that will be the sort of main area we need to focus on. Control of water. But we've then got regulation of salts and regulation of pH as other things that are going on through the through the microscopic structure of the kidney. You've got different parts that are responsible for these things. And then production of this hormone, EPO, we usually call it, erythropoietin, which stimulates blood cell production. So that's an overview. Now, in terms of the processes, we looked at these and recognised some, which is rather nice, isn't it? You know, when you see things that you've seen before, it does sort of ease the trauma a little bit. So active transport we'd already met, diffusion and osmosis we've already met, secretion to some extent we had, things like exocytosis, for example, and filtration. We'd sort of come across that when we were talking about tissue fluid, that it was the pressure difference, that high pressure going into capillaries would force out tissue fluid. So filtration's the one that's going to be really important to us first in the kidney. There is the edited version of that diagram. I copied it out, stuck it into paint, and I have edited this area of blood supply into the kidney over on the left kidney there, so that it should now match the diagram that you've got. All right, because the one that you've got is one that I edited. So we've got the renal artery going from the aorta into the kidney, the renal vein coming out of the kidney and back to the vena cava. So the blood supply in and out of a kidney is important, obviously, because the kidney's filtering blood. And then the product of the filtration process, the urine, is going to leave via the ureters. And of course, this diagram has this rather odd floating bladder down there as well that you don't have on yours. I can't even work out quite where it's supposed to connect up, because the ureters appear to be going behind the iliac artery and vein down here, which I'm not convinced they would do that. And actually, if you look over at the model there, they don't, do they? Can you see the ureters? And they come over the front, don't they? So if we were going to be accurate here, I think, you know, whoever edited this didn't do such a good job. That should have been coming across there, and that one across there. So I might edit that again at some point in the future, and all I have to do is pick up that colour and fit it in, just for the sake of accuracy, really. The next step then is to start looking at the ultrastructure of the kidney, in other words, the microscopic structure. So we're going to look now at the, oops, missed that bit. Oh no, we're not. We're going to look at the layers inside, aren't we? And that's the other one that you labelled. So on your blank diagram that doesn't have the blood supply, the bits that you needed, I removed the label from down there. The useful bits are the cortex being the outer layer, the medulla being the central layer, and the medulla is composed of these sections of tissue called pyramids. So they were the individual bits that look a bit like shells on the diagram, yeah? So the medulla is composed of pyramids, and then the pelvis here that leads into the ureter to drain the urine away. All right, so those are the important bits. You haven't got the artery and vein and arterial and venule and capillaries on your diagram, so it was really only labelling those bits. So we'll be back to those. The medulla is particularly where the osmoregulation bit goes on, so we'll definitely be coming back to that. The cortex, I've never really mentioned anything much happening there. So yeah, the medulla, the pyramid bits, and then the pelvis being the sort of drainage area. All right, so that must be as far as we've got, yeah, and at that point we abandon ship, and what we can now do then is move on to the microscopic structure. Now, if you take an individual kidney and look at it under a microscope, you see these tiny little units called nephrons, and each kidney's got about 1 million of these things. So if we take a small section of a pyramid, which is what's identified there, and enlarge it, you get these things over this side, and this is where we've got to actually focus our efforts. Right? It's the structure of these that's important. A nephron is a microscopic tube with blood vessels associated with it. All right, so that unit there is a nephron. Blood is supplied into the nephron. It does drain away, obviously. And then urine is drained away. So we've got a blood supply going in, urine coming out. And what we're now going to do is to look at the individual parts of the nephron. All right, this does get very complicated. There are a lot of component parts to it. But in your handout folders, you have got probably half a dozen different diagrams of a nephron. If you just flick through, you'll find you've got an unlabelled one, which unfortunately doesn't match the unlabelled one I've got on here, but never mind. You've then got several versions of it. Because I've put in about 3 different source materials, I think, you'll find all over them you've got nephrons. Yeah, nephrons everywhere. All right, so you know the words and things you'll be able to come back to and work out what they do. All right, this is complex, but it does quite nicely at the end, just ties back to the beginning of the unit with hormones and the nervous system. So yeah, I'm quite pleased with it actually. Okay, what's a nephron then? I've got several slides that go through this. First of all, looking at the component parts. The beginning bit is really sort of the focus of the first slide that follows this one. And then I've lumped the rest of them together, and then we'll look at trying to label the one that you've got. Okay, now this thing then is a nephron. Not an ideal diagram for nephron, but you know you'll start to recognise these as we go through them. There's 2 bits here. You've got the blood supply. You can see a lot of capillaries involved in this, and you've got the yellow bit on this diagram. The yellow bit is the kidney tubule. All right, now this is the bit that is going to contain the filtered out materials, what we call the filtrate, and what's ultimately going to be urine. All right, but it's not urine straight away, it has to be modified all the way along here. And I realised yesterday that this one is a slightly twisted version. Doesn't matter particularly. But wherever you've got one of these diagrams, if you can identify the tubule, and if you want to shade it in yellow, that will help you remember what goes through that bit. All right? So this bit, we've got the one to use later on. But we're going to label one, but anywhere where you've got one, the tubule can be coloured in yellow. It's the only opportunity we get to use the yellow pencils in the pots, I think. The blood supply on this one is not coloured ideally, because what we have with a nephron is a blood supply from an artery, or an arteriole actually, that comes in. It's still arterial blood when it wraps itself all around these capillaries round the tubule, but as it comes out, it's going to rejoin a vein or venule. So if, I would rather on this particular image that blood draining away was blue. Now, you won't necessarily be able to identify that on the diagrams that you've got, but you start, you're getting deoxygenated blood, obviously leaving the kidney. All right, but we've got a lot more to say about this yet. So let's start on the structural bits that we've got here. If you manage to label anything as you're going along, that's really good. I mean, use the diagrams we've got on here. Spread your diagrams out all over the desk or whatever, if you're trying to identify the bits, don't necessarily just work from the blank one that you've got. Pull some of the others out from the next few pages. Starting with the first couple of bits here. Now, one of the hardest words to say in biology is the first word on this slide. It's called the glomerulus. And for some reason, it's that R, that is the hard bit. It doesn't seem to fit glomerulus. That R sound in the middle there. So the glomerulus is, I have heard people try and get around it by calling it the glomerulus, which seems to be easier, but it's not. It is the glomerulus. And it's because we insist on having a U sound, I think. If we called it the glomerulus, and just an sound rather than a U sound, it would be easier. So glomerulus would be easier, but we call it the glomerulus. So it's a really odd one. So yeah, it's a microscopic blood capillary something. Right, I'm going to have to move the just briefly remove the recording panel across because I can't remember. Oh, right, a microscopic blood capillary bundle. All right, it's like a little sort of tangle almost of capillaries supplied by a branch from the renal artery. Okay, so we've got this little bundle of blood capillaries in the glomerulus. All right, so this thing here is this little tangle of blood capillaries. Now, wherever you've got one of those, you will have a blood vessel coming in and a blood vessel going out. They're both arterial blood. The one that's coming in is always described, this is not perfectly labelled on this one, but the one that goes into the glomerulus is known as the afferent arteriole. All right, so that one goes in. And then the one that comes out is the efferent arteriole. All right, would have been a better label. So if you can work that out on any of your diagrams, you're doing quite well. All right, we'll see how the diagrams go in a bit. All right, now, sorry? Zoom in on it? Not really, but I have got plenty of others, so, yeah? So at the level of the glomerulus, you've got a blood supply going in and a blood supply effectively coming out, because the efferent arteriole that comes out is then going to wrap itself around the rest of the capillary before it's drained by a vessel returning to the renal vein. Now, these capillaries round the tubule, we often call them peritubular capillaries, peri meaning outside, like perimeter. All right, the perimeter of a shape, or the perimeter, is measure outside, isn't it? All right, so perimeter. So peritubular means around the outside of the tubules. They then drain, actually, back to the renal vein. But that's a story for much further on. All I'm concentrating on at the moment is the glomerulus and the little cup round the outside of it that's called Bowman's capsule. All right, Bowman's capsule, it's a hollow cup-shaped structure, all right, and we're going to get substances forced out of the glomerulus into Bowman's capsule. The reason that we get substances forced out, this filtration process, and it's not obvious from this diagram or many of the others that you've got, is that the afferent arteriole going in is wider than the efferent arteriole coming out, so it puts the blood under pressure. Okay, so this one here should be wider than this one here. So that's our starting point for the whole process. That little bit, the glomerulus and the capsule around it, is sometimes called a renal corpuscle, but that's not a word you see very often. It's a bit of an old-fashioned one, so I don't use it. But if you come across it, that just check out what it is. Now, the rest of this structure is coming on the next slide, but with the same diagram. I thought I'll use the same one twice. But if you follow it, what we've got coming up now is, if we go from Bowman's capsule, we've got a wiggly bit, a loop that actually goes down that way, confusingly, because it's crossed over itself, and then another wiggly bit before it joins up with a fairly obvious collecting duct. So we've got wiggly bit, loop, wiggly bit, collecting duct. Now, of course, we're not going to call it the wiggly bit, are we? So it's got to be called the proximal convoluted tubule and the distal convoluted tubule. Yeah, so that's our next step, all right? So the next diagram, all right, giving us a clean version now. As you come away from Bowman's capsule, the first bit of tubule that you meet after that, so we go here, and so your diagrams will all be different shapes, all right, but that bit there is the proximal convoluted tubule, this thing. Proximal means, well, anyone come across that word before? Well, proximal, like approximate, near, or nearest, yeah? It approximates, or proximal. So proximal is actually the nearest end to the start, because we consider Bowman's capsule the start, so proximal is nearest. It's actually an anatomical word, and you can talk about proximal structures in your body anyway. Proximal, and then the other one you can see coming up is distal. So you would say, you know, your hand is the most distal part of your arm. Yeah, is the most distant, if you like. So we have the proximal convoluted tubule, usually, as you can see at the top there, just known as the PCT. Makes life easier. And we will talk about what goes on there. We've then got the loop of Henle. I'm not sure it's Henle or Henle, because it's actually German. Yeah. The efferent arteriole is the one that comes out of the glomerulus. This one, no, that's going to be a vein. This is the problem that we had on the last diagram. I've coloured this one in blue. So that's going to be the renal vein, ultimately. All right, so that's draining from here. Up at the top end, there's still arterioles. So, yeah, that should, I think, label there. I shouldn't have put blue around that, but it's too late. I've got it in my hand. And that's that one. Okay? Bit clearer? So, yeah, that, I mean, you haven't got this one, but this is a venule to keep our bits right on here. So, yeah, the loop of Henle. I'm trying to think of the guy's first, Friedrich Henle, I think. No? Yeah, whatever. German anatomist gave his name to this. I actually started making a bit more of an effort to you know, it's my attempt at diversity, is to name it, identify the nationality of the names. Yeah. So the loop is, well, all the diagrams you'll have after this will have it going, I shouldn't really use blue again, we'll have it going down and up, but the other way round, because this one, you can see there's a crossover there. It has what we call a descending limb and then an ascending limb. And the way that works is very complex, but it's about reabsorbing water. And it sets up what we call a countercurrent multiplier. But it's a useful thing to understand. Sorry, somebody mentioned fish. In fish, yeah, can you remember whereabouts in a fish? Sorry? Fish don't have lungs, gills, yes, you're absolutely right. And that's, yeah. Some fish have a countercurrent multiplier in the gills because they have a bit of an issue getting enough oxygen into their blood. So they have blood flowing one way, water flowing the other way, and it maintains a concentration gradient across the surface. Can you remember which sort of fish? It's the bony fish. The other sorts of fish, the cartilaginous fish like sharks, have the blood and the water flowing the same way, so they're much less efficient at getting oxygen. So, no. You tell that to the next shark you encounter. Yeah. Once we've gone through the loop of Henle, we then have the other wiggly bit. So we've got the distal convoluted tubule here. This is rather a pathetic little one. And then that all joins up to the collecting duct at the end. All right. Now, the bits we're going to focus on, what goes on in Bowman's capsule, which is the filtration bit. We're going to look at selective reabsorption in the PCT, all the water control in the loop, and then after that I'm just going to abandon it basically because the rest is more than we need. Okay, so those are the parts anyway. Now, this diagram is not the one that you've got, is it? I sort of skimmed through diagrams and thought, oh, that's the one they've got, because they always look the same. That, however, is a labelled version of it, which might help you to label the one that you've got, if you've got any, sorry, get that out of the way, any bits left on your diagram that need labelling. I had to actually redraw the one that you've got in your handouts for the other group because this threw them completely. But I think I've talked it through a bit better this time, so hopefully you've got the, you know, the narration of the parts as I've gone through. See, I learn from my mistakes. So what we have, you will have up at the top for your glomerulus, wherever that is. Hang on, let's check. It does at least go in the same direction, I think, doesn't it? Yeah, not quite. Yeah, yeah. No, it doesn't. Yes, it does. Sorry, it's yeah, yeah. Where you've got blood comes into your glomerulus from the top. All right, so that is your top blood vessel. It's the equivalent of that bit. Your glomerulus is then your tangle of blood vessels. So this one that I've got on here is better actually than the one you've got, isn't it? Because it does show the efferent arteriole going in being wider than that one, the afferent. So I've done that back to front, haven't I? Afferent goes in, efferent comes out. So it's actually showing the afferent being wider than the efferent. So yeah, E for exit or entrance. So we had this. A was arrive, we decided yesterday. So A for arrive, E for exit. So we've got the branch of the renal artery coming in. That's an arteriole by the time it gets there. You've got the afferent arteriole going into your capillary network, into the glomerulus. So that's from the top. And then your efferent one coming out. Have you got that identified all right? You happy with that? That efferent arteriole then is the one that comes out and wraps itself around the rest of the tubule. So where, you know, you can follow yours as far as you like, sort of around here, all wrapped about. This is still branches coming from the efferent arteriole. Okay, but they are capillaries at this point. Okay, I'm not going to colour them all in, but you know, feel free if you want to. So there, you know, wrapped around that loop, they are still oxygenated blood, but they will start to become deoxygenated. So if you can make them a sort of purpley colour by the time they leave, and then connect them up to the renal vein in blue, then you've cracked it, all right? But that's, yeah. Yeah, a venule is a small vein, a branch of a small vein. So you've got, yeah, a branch of the renal vein will cover that. And then, yes, you can then probably work out how much of that can be coloured red and blue. And then yours has got quite nice wiggly bits, hasn't it? So if you start at the cup-shaped bit, you've got Bowman's capsule there. And I can't use yellow on this one, I'll have to use green. Green urine is never a good sign. All right, but that's your Bowman's capsule then. And then follow from Bowman's capsule and you come down to your proximal convoluted tubule there. All right, so you've got, yeah, you've got a decent little proximal convoluted tubule that goes up and down, haven't you? It comes down into the loop. The loop that you've got is better than this one because it shows different thicknesses, and that's partially associated with the way it works. You've got a thick bit, a thin bit, and then another thick bit on yours. So you've got the loop of Henle, goes down and then up. Okay, so that's better than the one I was using. And we can identify, I've got more label lines on here than I need. Or than have been used, haven't I? Yeah. This, where you go from Bowman's capsule through the PCT, the proximal convoluted tubule, down this side, there is a label line that I could use here. That first part, you don't need to put the label on at this point because we will come back to it, but it's known as the descending limb of the loop. All right, so to put that in brackets, if you've got space, and then, all right, so because the filtrate is coming down here, and then it comes up the other side, so not surprisingly, this is the ascending limb. But we will deal with the loop in its own right, so we'll definitely be coming back to those. At the moment, it's just an overview. So then continue on from your loop, and you come to the other wiggly bit, your distal convoluted tubule, and then the collecting duct down there. All right, so those are the important bits. Okay, so that's what we would call kidney ultrastructure. All right, the rest of it is the sort of gross structure, the large-scale structure. So we've now got the ultrastructure bit. So to get your head around those words, because there's a lot of new stuff there, and these, you might have heard of parts of the heart and the lungs and the brain and stuff like that before, but the kidney tends to be new to everybody. It's not something that you really talk about the structure of it on a daily basis, do you? So we're going to do a little recap now. So I'm going to give you these diagrams to colour if you feel that way inclined. They are actually quite useful because they just give you the parts and letters, and then you colour the name of the part and the part itself in matching colours. And if this works for you, then you can look for some of the colouring exercises for revision. So it's what we're working up to as revision techniques. Also going to give you the tarsia puzzles to play with, so we'll put this on pause for now. In fact, I think I'll stop it and record this at this point, or save it at this point.